I am Bethany Grant, CTO at Kytopen, and I am going to talk to you about unlocking the potential of cells with the FlowFX system. Let me start with a question. What would it take to change the current state of immunotherapy production and access to state-of-the-art cell therapy-based care? What if personalized cures for cancer and genetic diseases could be developed, discovered, and delivered as a first line of care in time to save lives and ultimately in order to prevent disease? Solutions to enable personalized cures require significant change. To get from manual, expensive processes that treat only the sickest patients to efficient, affordable, and personalized but widely available solutions takes transformational change. This requires innovation, not merely incremental change. We're all familiar with the complexity of today's manufacturing process. I won't go through the specific details here, and this is showing a viral-based process, but this demonstrates a process requiring many steps with a variety of tools and separated from the patient. Feeding into this end result is that discovery happens with people, processes, tools, and organizations separated from process development. Many disconnected manual tools are put together to comprise a full process. But what if we started with the patient at the center of this problem, so that discovery focused on the pathology, process development considered the specific challenges of the disease, and the material manufacturing could solve each individual's challenge, but still be scaled? What is needed to fully realize this solution? Well, what if discovery, process development, and manufacturing used the same tools? What if a batch was limited only by the patient material? And what if a process could be optimized by the tools available instead of optimized around the available tools? What if the patient was at the center of the process? How do we do this? Well, we at Kytopen can improve upon the current tools and processes, inching closer to transformation by connecting discovery, process development, and manufacturing. But we can't do it alone. We also need to partner with other agents of change. So how are we doing it? Kytopen has developed a system configured in a small volume, arrayed high throughput flow effect array, and a large volume closed system for GMP manufacturing, using the same transfection delivery system. This solution was specifically developed to address the problems identified earlier and based on customer input. This platform system enables discovery in the array and then seamless experimentation, evaluation, and parameter optimization through the process and confirmation at large scale at any stage. As manufacturing matures, the need for the TX manufacturing system increases, and the need for the small-scale development array decreases. But even through lifecycle management, studies can easily be performed at small scale. The FlowFX system takes advantage of electrical, mechanical, and chemical kinetics to induce pore formation in cells and deliver endogenous material through endocytosis. No other technology utilizes so many factors, enabling Kytopen to optimize for high efficiency of delivery while maintaining cell health. We've demonstrated delivery in a wide and expanding range of cell types and payload materials. Early development of the system utilized GFP mRNA in a variety of cell types, including the ones you see here. But current focus includes more clinically relevant payloads, targets, and cells, including CRISPR-RNP and nuclease mRNA, knockout in T cells, NK cells, CD34 stem cells, and as well as a focused effort currently underway on knock-in with various payload constructs. Here's data demonstrating delivery of GFP mRNA with the FlowFX system in multiple cell types, including T cells, CD34 stem cells, and B cells. both on the small volume array and the large volume TX. Viability greater than 85% was achieved, with efficiency greater than 90% on both the small and large systems. We're able to explore a wide range of electrical, chemical, and mechanical parameters in order to define optimal settings. In this case, we looked at RMP knockout of TIGIT and primary human T cells. Let me take a few minutes to explain what we're showing here. We completed over 300 experiments, normalizing results to a reference setting, which you see in X right here. On the y-axis, you see editing efficiency measured in % indel of TIGIT. On the x-axis is normalized total viable cells. We plotted the results and also included in color markers the amount of electrical energy delivered at each setting. We also included a static electroporation as a control. That's the triangle marker here. We found settings on the FlowFX system that perform as well or better than static EP. Also notable is that we're able to achieve those results with lower electrical energy. Given that we have more factors to consider with the electromechanical chemical optimization, we use a multi-objective weighting system to identify optimal settings. We selected 3 of the best FlowFX settings shown here in the rectangle, and noted we're only plotting 3 parameters, although we have many more that we fully optimize. It's just difficult to plot on a 3D plot. And we also included 1 suboptimal setting identified, including the optimization effort, so that we could demonstrate the replicability of the system. We also included a commercial static electroporation system control, and confirmation runs were completed for 3 donors. The best 3 FlowFX settings are in blue and the static EP are in red. We saw improved viability and growth as measured by population doubling level in for all 3 donors, as well as maintaining high comparable efficiency, editing efficiency in all 3. So manufacturing is complicated today. More all-in-one systems are becoming available, and in the future, manufacturing will come to the patient. Because of the consistency in delivery at small and large volume, coupled with the continuous flow, FlowFX does not have the batch limitations typical to static EP or other non-viral solutions, and is perfectly suited to improve problems with current manufacturing and integrate to enable tomorrow's solutions. Kytopen is executing development plans on both the TX for GMP applications, with evaluations ongoing, and also the array for research use. We're happy to consider interest in evaluating a device or discussions in areas of strategic partnerships, including an integration into an all-in-one manufacturing system, acceleration of the array for discovery or process development, or payload delivery optimization for a clinical application. Kytopen is led by a strong team, including its 2 co-founders from MIT, as well as great financial and in-kind support from notable investors and organizations. Although we've doubled our team since this photo was taken last fall, we have a great team supporting development, operations, and commercial activities. Thank you very much, and please reach out if you have questions.